Hi there, I'm Russ Altman from Stanford University. I'm a professor of bioengineering, genetics, medicine, and computer science. Today we're going to talk about a regulatory science research roadmap for next-generation sequencing. This is a critical and emerging area. So the Precision Medicine Initiative, or PMI, is a new initiative instigated by the President Obama, and announced in the State of the Union address in 2014. It involves principally a 1 million person cohort of Americans that are going to be followed over time. We're going to study their genetics, their lifestyle, their environment, the medical records they have, and even data from wearables in order to understand how to more precisely diagnose and treat disease. So of course NIH has a huge role in doing this research, but it's expected that a lot of this research will lead to new technologies, new treatments, new diagnostics. And that's where the FDA comes in. The FDA has an important role in figuring out how is it going to manage these new inventions, bringing them to market, making sure they're safe for the American public. They've created a program called Precision FDA. It's actually precision.fda.gov, which is a portal where people can share data about next-generation sequencing and start to develop the scientific basis for evaluating the accuracy and utility of these new tests. A lot of the work in Precision FDA and in the new diagnostics happens at the Center for Devices and Radiological Health, one of many centers at the FDA, CDRH. And in particular, the Office of In Vitro Diagnostics. Next-generation sequencing is very often considered a diagnostic. It's in vitro because it's happening in a beaker. And so the Office of In Vitro Diagnostics is the place where a lot of the activity for next-generation sequencing happens. In September of 2015, members of the CDRH Office of In Vitro Diagnostics came to Stanford as part of the UCSF-Stanford Center of Excellence for Regulatory Science and Innovation, also known as CIRSE, to have a discussion, public discussions and private discussions, about the research knowledge that would be required in order for the FDA to make the best decisions about next-generation sequencing-based diagnostics. It was a successful visit, and out of it came many initiatives and collaborations. One of the collaborations was published in the journal Science Translational Medicine in late April of 2016, and it was a paper with a title that I hope you'll recognize because of today's lecture, A Research Roadmap for Next-Generation Sequencing Informatics. These were folks from academia, industry, and government who in this 3 or 4 page article, which I hope you have access to, outlined 9 areas that require research in order for the FDA to have the knowledge base it needs to make the best decisions about these new technologies. I mentioned before that the Precision FDA website, precision.fda.gov, was set up just around this time. There's an FDA contract to a company called DNA Nexus that is building this portal. It provides a framework for data and software sharing. It's a research tool. At the FDA, they distinguish between research tools, which are meant for the discovery of knowledge relevant to regulation, versus a regulatory tool, which would be used by the decision makers at the FDA to actually evaluate technologies. Precision FDA is specifically a research tool, not a regulatory tool. The idea is that it will inform the regulatory tools that may be built later on based on what we learn in using it. It's available to credentialed researchers who can store genomes there, submit software, and share with one another. And its vision is really to create kind of a sandbox for people to test new ideas, establish benchmarks. We'll talk about this in detail in a few moments. Participate in challenge problems where somebody says, hey, I have this file. Who can do the best job at analyzing it? And just generally understand the issues related to the interpretation of next-gen sequencing data, or NGS. I'll be using those interchangeably. So why are NGS tests different from traditional tests? The FDA has a long history of looking at diagnostics. First of all, and I think most importantly, a next-generation sequencing test can simultaneously make millions of measurements on a genome. So it's not focused at asking one question in general. The human genome has 3 billion DNA bases, and in principle, a next-generation sequencer can query all 3 billion of those bases. That can create problems because some measurements are interpretable today. We know at a certain position in a certain chromosome, if there's based on what we see in the DNA, we know how to interpret that for disease risk or drug response. However, many measurements are not currently interpretable, and yet we're measuring them. That causes a problem. What are our responsibilities to manage information that we don't yet know how to interpret? The third point here is that some measurements are not interpretable today, but will become interpretable in the years to follow, or months or weeks. What is our requirements for revisiting data sets to see if there's anything newly interpretable that wasn't interpretable before? Of course, different technologies for sequencing have different error models. What I mean by error model is the types of errors they make are often very different. We need to understand those and characterize them. And different technologies are, because of these features, best suited to different uses. But we need to be able to write down which technologies are particularly well suited to which kinds of measurements. These things combine to make NGS a special challenge for regulation. And yet regulation is critical because without a clear regulatory pathway, many startups and companies creating very exciting new diagnostics won't know how to interact with the FDA and won't know what the expectations are of the FDA for the kinds of evidence they need to submit in order to get their products approved for general dissemination. And that could limit the penetration of these valuable technologies into routine medical care. So before we get started, I want to just remind you of some of the basic key features and definitions that we'll be using about diagnostic tests. So every diagnostic test has an expected use. This is typically the clinical, although it may be a research goal that is being pursued when you're performing the test. Or informally, why is this test being ordered, either by a clinician or a researcher? How are you going to use this information to make a clinical or research decision? Once you have that defined, the next question is, what is the interrogated range of the test? Some tests, in fact very few tests, require the entire genome. Sometimes we're just interested in a specific letter of the 3 billion letters in the genome, A, C, T, or G, at a specific position. Sometimes we're looking at an entire gene. Sometimes we're looking at a segment of a chromosome. We have to be able to define the region of DNA that's of interest for this test, and we call that the interrogated range, the portion of the genome being examined. It can range, as I said, from a SNP to an entire chromosome to an entire genome. Within that interrogated range, you're interested in knowing what kinds of variation in the DNA is detectable or would you like to detect. Are you just looking for single letter changes from an A to a T, from a C to a G? That's a single nucleotide polymorphism, or SNP. Are you looking for insertions and deletions, or indels? If you're interested in insertions and deletions, are you looking for a single letter deletion or insertion, or 2, or 5, or 500, or 5,000? There are different characteristics of different technologies in terms of their ability to measure small indels versus big indels. And then, of course, we might be interested in copy number variation. These are entire genes that are copied throughout the genome more than once. And then large-scale structural rearrangements where big pieces of DNA have been shuffled. All of these have potential applications in different disease settings, but when you're just evaluating a test, you have to say, for the interrogated range that you're looking at, which kind of variants are of interest. Okay. From that paper that I showed you, this is table 1 that gives some examples of expected uses. That was the first thing we defined in the previous slide. One kind of test might be non-invasive fetal testing, and the use there would be to look for chromosomal abnormalities that might be associated with harmful or even lethal phenotypes in young babies. That's different from targeting gene panels for a cancer tumor, where what you're looking for there perhaps are mutations in the genome that might be driving the cancer, might be feeding the cancer, and which would therefore be very attractive as drug targets. You might be doing a carrier test for somebody who's thinking about having a baby. And there you might be looking for, the question is, does the individual carry a known or a new mutation that might impact the health of the child? You might be looking at a whole genome to try to understand if a child has a congenital disease or some condition, what its cause is. There's this idea of a diagnostic odyssey, a child who seems to be not thriving. There's a suspicion that it's a genetic abnormality, but we don't know what it is. So you sequence the parents, you sequence the child, and you look for variations that might explain what's wrong with the child. We can do whole genome sequencing on cancers, especially in the emerging area of immuno-oncology with immune therapy. And we might do whole genome sequencing to calculate the risk of an individual for common diseases like diabetes, schizophrenia, depression, hypertension. All of these are different expected uses and would change your tolerance for error, the types of variants you might look for, and the approach to testing. And you can't evaluate a technology unless these things are defined. Otherwise, it's an ill-defined evaluation problem. So in this paper that we wrote, we identified 9 areas that represent 9 general research domains that require the attention of science in order to help create the knowledge base that will inform FDA regulators as they make decisions about these technologies. And we're going to go through them in detail, but they are secure storage of genomic data and software, reference data sets covering expected uses, error models of the different sequencing technologies, methods for comparing different software modules, best methods for creating test data sets, understanding population genetic effects, both on the detection and interpretation of variations, understanding technology performance under certain conditions and contexts. We'll go into all of these in detail. Methods for using non-FDA databases for making regulatory decisions. And number 9 is understanding how to communicate all of this complex information to providers and to their patients so that we can understand how to use this most effectively. So now what I'd like to do is just give you some highlight issues in each of these 9 areas. First one is secure storage of genomic data and software. First of all, we need to be able to take this data and we make sure that we respect the privacy of individual genomes, either because they're patients or because they're study subjects who have an expectation of privacy. Can these be re-identified? Even if I take away the name and address and social security number of a patient, it's possible that their DNA could be used and linked to other databases to figure out who they are. There's an expectation among patients and of research participants that this won't happen without their permission, and so we need to have methods for robustly guaranteeing as much privacy as possible and disclosing to patients when things are not as private as we'd like. We need to manage very large volumes of data. The genome, as I said, 3 billion bases. The raw data files can be terabytes of data. We have to have standard formats for exchanging information. We need to be able to make sure that when we use the genome of an individual, that we are allowed to use it based on the consent that they signed. So there's a complex set of consents, samples, and what you're allowed to use for what kinds of purposes. Some participants in research only want their DNA used for example, the study of diabetes, but they do not want it used in the study of hypertension. And we have to respect those wishes. Searching and retrieving information from this very large volume of data, both on individuals. Have I ever seen an individual with this particular DNA variation versus how many people worldwide do we expect to have this variation? Need to be able to answer those kinds of queries. We need ways to share software and yet respect the intellectual property of the people who created the software. A non-trivial problem, but in the setting of Precision FDA where people are sharing their software, they need assurances that their data is safe and their software is safe. Very interesting issue is version control. In many disciplines, if you have a piece of software and you discover a bug, you fix the bug and you do a new release, and on your iPhone you're updating your apps all the time because they've made improvements. How about this, however? If the FDA has approved a previous version of the software for a particular use, it is not obvious that you can just change that software and release a new version. The FDA had specific tests and expectations on that first version. The new version you may think is better, but we need to have a system to make sure that you haven't introduced, in changing the software, unexpected features that make it less good or even harmful to patients. So this issue of the version controlling of analysis software is a big one. Finally, I want to mention the Global Alliance for Genomics and Health. It's a public-private partnership working on many, but not all, of these issues. And in the next slide, I show the website for the Global Alliance. And they have this Beacon project, which is about one of the challenges. The Beacon project is devoted to allowing different sites to tell users whether or not they have any samples that have a particular variation of interest. And the idea is there are little beacons. You can see the picture on the far right that are just broadcasting features of their database so that people can determine if they're of interest. OK. Second thing on our roadmap is the creation of reference data sets. We will be accumulating a large and diverse set of sample genomes. We want to make sure that these genomes represent the American public. We want to make sure that they cover all the ancestral populations of interest, which is basically all human ancestral populations. And to make things more complicated, humans don't all come from the same ancestral population. They have parents and grandparents from all over the world, and so they are admixed. We need to have samples of all of the above to ensure that the tests that we run on these technologies are fairly representing the diversity of humanity. A somewhat specialized concept within genetics, maybe familiar to you, is linkage disequilibrium, which refers to the correlation structure of variation. Without going into too much detail, if you have a variant in a certain position, it might give you information about the likelihood that you have a different variant at a different position. That information is very useful for making diagnoses because sometimes you can measure this and make a lot of inferences about other positions in the genome. However, that linkage disequilibrium pattern can sometimes be specific to different populations. And so we really need to understand the degree to which one variant can predict another variant. We also need to make sure that we can asymptotically have very high quality genomes where we know exactly every base at every position for the purposes of evaluating new technologies. This is not trivial because even the best current sequencing technologies have certain blind spots in how they measure things and therefore don't have a perfect picture of the genomes that they're applied to. There's a very important initiative run out of the National Institute of Standards and Technologies, or NIST, called Genome in a Bottle, GIAB. It's a collaboration that is trying very hard to create samples that are very well characterized down to as well as we can do it at every single base so that we can have literally a little tiny vial that has a genome in it that can be sent to a company blind and say, sequence this genome, tell us what you find. That will allow us to see how accurate your technology is. So in this slide you see in the upper left, literally one of these little test tubes that NIST will send to companies. This says human DNA for whole genome variant assessment. Very cool, and you can expect to have libraries of these in the future, as I said before, covering many ethnicities and many admixed populations. This is the genome in a bottle. In the lower left, you see the genome in a bottle logo. It's a bottle with a picture of a genome on it. And then on the right, you can see the general flow from input data sets to a best guess, and it's not a guess, a best estimate of the genome that is then put into a bottle. So a very exciting, and NIST and FDA work together on these types of projects. Number 3 is error models. Every sequencing technology makes different types of errors. These are very complex, high throughput technologies. They involve light, they involve photons, they sometimes involve measurement of very rare events. And based on each company's technology, there's strengths and weaknesses. We would like to have software to model the strengths and weaknesses of each of these technologies so we can have a computational model of how the errors might affect different expected uses of sequencing technologies. So we'd like to know, does the technology match the interrogated range that it claims to match? What kind of variations can it reliably measure? And therefore, which expected uses might it be well suited for? There are many companies working on novel DNA sequencing technologies, and we expect them to have very different parameters in this space. And as I say in the third point, we must evaluate their claims about accuracy and coverage. It's interesting also to think that sometimes the best way to get a genome sequenced will be to use multiple technologies, especially if two technologies have complementary errors. One technology makes errors of a certain kind, the other doesn't make that errors, and therefore by bringing them together, you get a much more accurate genome. There is a tool called ART that allows you to build error model abstractions, and so that's an exciting tool in this space which is just beginning to grow. Here's the website at the National Institute of Environmental Health Sciences where you can download ART and simulate errors in your favorite genome and then see how important those errors are for your expected use. Number 4 is to evaluate and compare pipelines. Virtually every company and virtually every academic group working in genomics has created its own software pipeline that takes the raw data and comes up with a call of what variants are present in the genome. Even for these single technologies, we have like Illumina, which is one of the companies that does sequencing. There are many different pipelines that can analyze that data. We need ways to compare these pipelines and figure out who's doing well and who perhaps is doing less well. And so one of the ways to do this is to have objective comparisons in a public forum with a known answer, known to the evaluators, but perhaps not known to the participants, at least initially. Send it out, have them all do their best, bring it back, and see with the exact same input data, how do these different pipelines perform. We need to figure out how to score these so it's fair. I don't expect this will be a single number. I think this will be a set of numbers that evaluate each pipeline along a number of criteria. We have to remember about systematic bias coming from assumptions about how DNA sequencing will work. There are a few vendors currently that are dominant. They may be dominant next year, or they may not. We have to make sure that our tools are general purpose and can handle anything that comes down the pipe, so to speak. And finally, we need to make all of this available so that vendors who are coming up into the field have ways to evaluate the real performance of their technologies on unbiased data sets. So the evaluation and comparison of pipelines is critical. The Precision FDA site currently, at this moment, May of 2016, has 2 challenges that have been announced where there are many participants around the world taking these data sets and submitting their data for comparison and evaluation. So that's a great step. The 5th of the 9, so we're a little bit more than halfway, is the creation of suites of benchmark data sets. The creation of gold standards is not trivial for next generation sequencing. And I'd like to kind of have you think with me about what the choices are. One obvious choice is to create synthetic data sets. That is to say, a computer program creates a pretend genome that can be then fed into a pipeline. The advantage of that is you know the answer because you synthesized in a computer that exact sequence. The problem is it might not be realistic. Real genomes are complex, and we don't probably know all the ways in which they're complex. So if we build a synthetic genome, it probably will be missing some of the features that a real genome would have. So you might be falsely confident in performance because you're only showing that you can measure what you know about. So then you go to the other side, which is, well, maybe we should only take a real genome, like the Genome in the Bottle project. The problem there is this is extremely realistic data. It's an actual human genome. However, we still don't fully understand the structure and function of the genome, so there may be elements of it that we haven't quite measured or sequenced accurately. So a new technology may actually get it right, and we might not recognize that they've gotten it right because they're the first technology who's been able to measure this particular feature well. So then in the middle, you have this idea of let's take a real data set but inject synthetic data into it. So we have the real DNA reads from a real sequencing run, but then with the computer we're going to pretend like we had other reads that have been synthesized. Some people like this because it's somewhat realistic. It has real DNA, real genomic DNA as part of the data set, but at least for parts of the data, you know the exact answer because you put them there. So it's kind of a compromise. Some would say it's a great compromise. Some would say it has the worst features of both of those extremes. So we need research to understand how to do this. We need to make sure that we uncover all the interrogated regions, the types of variation, and the expected uses, because it may be a complex answer. And there are some tools available that are able to create, for example, synthetic data. I point to, in particular, VarSim and the Hive in silico projects that are available via Google. Number 6, characterize population genetic effects. I referred to this briefly earlier. It is critical that the PMI look at underrepresented and understudied populations that have traditionally perhaps been ignored. These populations will have a different genetic architecture in terms of the detailed, for example, linkage disequilibrium than other populations. And so in the same way that we have to have samples from those populations, we also have to make sure that we understand how to analyze and interpret measurements made on those populations. It's critical because sometimes we might have a variation that is associated with some phenotype of interest, like disease risk or drug response. That's very different from being causal for that phenotype. So I'd like to highlight the difference between correlation of a variant with a phenotype versus causation. Causation you can often just take to the bank, and it's likely to be true across all populations. The statistical association can be very different across populations. As I've mentioned before, we have to worry about all the ancestral populations on Earth and all the ways that humans can mix together to create new humans. We probably have to think about ethnicity-specific reference genomes. There are some genetic variants that are very common in one population, but very rare in another. And so your degree of surprise when seeing that kind of measurement would differ across populations, and it might affect the way you do your interpretations. And then, of course, this is one of the major goals of the Precision Medicine Initiative, and so the FDA and those of us interested in regulatory science will be coordinating with the researchers who are part of the Precision Medicine Initiative. Here's just a picture on the left of a particular SNP, single nucleotide polymorphism, a single letter change. This is actually a change from A to G. In Africa, where all humans arose, everybody virtually has an A. But as you see, the migration of populations to Europe, in particular, this G allele became more common. And meanwhile, in the Far East, it's all still A, and there's kind of an intermediate frequency in the Near East. These are the kinds of distributions that need to be understood for every single variant in the genome, and it's a non-trivial problem. On the lower right, we have a linkage disequilibrium map. I won't go into too much detail, but the bright red indicates SNPs that are highly correlated. So in the very bottom, at the point of the triangle, there's a bright red box which tells you that a SNP on the far right and a SNP on the far left are highly correlated, and it's almost, in this population, unnecessary to measure both, because once you measure one, you could predict the other. That map, however, is not going to be generally true for all humans, and so we need to understand the linkage disequilibrium map for each population. Number 7 is to evaluate the context fit for these technologies. There are regions of the genome that are well known to be difficult to sequence. Just two of my favorites are the human leukocyte antigen locus, or HLA locus, very important for the immune system, very difficult for existing next-gen sequencing technologies, or at least some of those technologies, to get right. Similarly, drug metabolism is in a large part determined by the cytochrome P450 system, and those genes are notoriously difficult because of their close relationships, which make it difficult to tell if you're sequencing gene 1, gene 2, or gene 3. And there are others. These difficult regions, unfortunately, are medically incredibly important. So first of all, we have to understand which genes are going to be easy and which genes are going to be hard. And we need to understand easy and hard for which technologies. That mapping will help the FDA look at new submissions and say, oh, they're using their technology for something that they're very well suited for, versus, oh, they're using their technology on something that traditionally hasn't been one of their strengths, so let's have increased vigilance in taking a look at this submission, seeing how good the data looks. So we need to evaluate technologies on these particularly challenging areas, and we have to consider how to use external data resources to support, first of all, to define what's the medically important regions, and then to support the idea that some of them are easy and some of them are hard. So this context and fit of technologies to different types of important genomic areas is a big question. Getting near the end, number 8, using non-FDA data for decision making. Many non-FDA databases. When I say non-FDA database, I mean a database created by a company or by an academic, available to the world or for sale to the world, but not under the control of the FDA. Although the FDA doesn't control it, it still may be absolutely critical for FDA scientists to use in making decisions about products in the space of next-generation sequencing. How should these products be used by the FDA? Some examples are the databases ClinVar, ClinGen, PharmGKB, brought to you by Stanford University, OMIM and HGMD, and many others. These all have information that could be of interest to the FDA, but there's a question. Where does the data come from? How high is the quality? Do the people who create these databases routinely abide by standard operating procedures to make sure that the data is all good? This has to be documented because if the FDA is going to use this information to make decisions about products that will affect patients, they are intensely interested in the quality of these databases. So although they don't control them, they need to qualify them for use in regulatory decision making. FDA therefore has to declare which databases it has evaluated and will use. The people submitting new tests need to know which databases they might be put up against. And this allows a focus, again, on the appropriate interrogated ranges, types of variations that are measured, and expected clinical or research uses. And then, as I mentioned for versioning before, these databases are all actively updated. So if the FDA likes it in January and it gets updated in February, does the FDA still like it? And what are the procedures required to make sure that the database is still reliable? This is the ClinVar database. I'm sure you can't read this, but it's the entry for a gene called NOS2. And ClinVar is a database of clinically important genetic variation. And this particular entry tells you that there are certain variations in this NOS2 gene that are actually protective for malaria. So people with those variants have lower risk of catching malaria. One of many hundreds of entries in the ClinVar database. This is ClinGen, which is a resource for clinical genetics. And this is the same gene, NOS2. And you see a variety of hits, including places where you can buy genetic tests that test this gene, information in PharmGKB, a database for pharmacogenomics, and others. And then finally, I have a snapshot from the PharmGKB itself. This is for a different gene, interferon. And this, again, we're not going to go into detail, but what this lists are variants in the gene, some of their synonyms, and some of the drugs whose response is affected by those variants. All of these are potentially interesting and useful to the FDA, but how can the FDA be sure that the quality is good enough to make regulatory decisions using these data? And finally, developing methods for communicating the results. Studies show, many studies, that providers, physicians, and others are very interested in using genetics in their practice. However, very few of them feel perfectly well prepared to use genetic information. It just was not part of their training. It's not part of their daily workflow. So, of course, professional education is key. And patients increasingly want to participate in their care, and so it's not just about explaining it to the doctors, it's also about explaining it to the patients. And in fact, helping the doctors and nurses and genetic counselors explain things to patients is critical. Genetic concepts, almost needless to say, can be very complex, and yet in these patient interactions, clinical providers have a limited amount of time, and so they need to have good ways to communicate the risk that is seen in a genetic test and what it means for the patient's health. There are some good examples, although we need more, of publicly available resources. And what I'd like to point out is the Genetics Home Reference brought to you by NIH. In addition, there is increasing interest at the FDA in understanding patient preferences for how they would like to receive information, and also patient-reported outcomes. Did this genetic test serve your health and your interests well or not? A very active and growing area of FDA research. Here we just have a screenshot of a page at the National Library of Medicine for the Genetics Home Reference, and it discusses a particular gene, thiopurine S-methyltransferase deficiency, and in a fairly easy to understand, not meant for professionals way, it describes the clinical syndromes associated with this genetic variation, whether there are treatments, and what it means for the health of the patient. This kind of work needs to be now made routine, and the FDA needs to think about its relationship to such summaries. Do they regulate these summaries? Do they read them and use them in their own decision making? A very important question. So we've just gone through 9 areas in the research roadmap. I will be the first to say we didn't solve any of those 9 problems, but we understand what they are, and it's a challenging roadmap. Progress on any of these 9 areas is critical for accelerating the implementation of next-generation sequencing diagnostics into the clinic. And ultimately, it's our assumption that if this is done well, patients will benefit. The precision.fda.gov site is part of this precision medicine initiative, and it's devoted to helping catalyze the research required for the FDA to make better decisions. This is something more than the FDA can do alone, and therefore the paper that we wrote was kind of a call to arms, if you will, for the entire research community to take their part in addressing these challenges in order to accelerate progress. There will be ongoing challenges as new technologies emerge and our ability to interpret genomic variation improves. So I want to thank you for your attention. This lecture was brought to you by the UCSF-Stanford Center for Excellence. My email is there. Thank you very much.